Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts of biosciences. Today we are going to deal with the topic oligosaccharides. Oligosaccharides are when monomeric units of monosaccharides, about 2 to 10 monosaccharides units, when we join them together, we will get an oligosaccharide. So if there are 2 monomeric units, we will get a disaccharide. If there are 3 monomeric units, we will get a trisaccharide, tetrasaccharides, pentasaccharides, etc. And all these are available in nature. Most of these are available in nature. And they have varieties of functions to perform. So let us primarily see how these oligosaccharides are made. So therefore, if you look at the structure of sugar, in the structure of sugars, let us represent that based on the functional groups. So therefore, based on the functional group, let us represent it this way. This is an example of an aldehyde. So what happens to an aldehyde is, add ROH. So what happens is, this aldehyde, it becomes O, and this gets distributed. Okay, so this is what we called a hemiacetal. Remember, a hemiacetal is formed because of intramolecular rearrangement. Suppose we add another ROH, what will happen is, so therefore, this will become R. Oh, sorry, here there is a hydrogen, O, OR, and this becomes OR. Okay, this is what we call an acetal. So therefore, due to this rearrangement, we will get an acetal ring, acetal bond, or a glycosidic bond in the case of a monosaccharide. So when 2 monosaccharides unit comes together, it forms a glycosidic bond with the loss of a molecule of water. So therefore, each of the monomeric units in a disaccharide or in a trisaccharide is what we call a residue. Remember that. So this is the basic mechanism. And the same thing can be applied if it were a ketose. Okay, if this were a ketose, what change we will introduce? So if this is a ketone, so therefore this is going to be an R. So the only change will be, this is R and this will be R. This is the only change. You will get instead of hemiacetal, it becomes a hemiketal. Hemiketal, remember, it is an old terminology, and similarly it will become a ketal. Okay, so ketal is an old terminology. Just to acquaint you, using the terminology hemiketal and a ketal. So this is how disaccharides or trisaccharides are formed, by forming glycosidic bonds. This is the basic that we need to understand. As you know, the oligosaccharides, they are like other carbohydrates. They are crystalline, almost white in color. They are soluble in water. They are sweet. So these are the physical properties of oligosaccharides. Now, these oligosaccharides have varieties of functions to be carried out. The most important function of these oligosaccharides is to give the energy-rich molecule. So therefore, they give energy. That is one of the functions of oligosaccharides. But this is not the only function of oligosaccharides. There are oligosaccharides carrying out other very important functions. For example, there is an oligosaccharide which is present in the gram-negative bacteria. And this oligosaccharide is made of about 6 to 12 glucose unit. GLC is a short form of glucose. It is made up of 6 to 12 glucose unit. And they are all formed by beta 1,2 linkage. Okay. They are formed by beta 1,2 linkage. Now, what is the function of this oligosaccharide? This oligosaccharide in the membrane of the, if this is the membrane of the bacteria, this oligosaccharide is present here. This oligosaccharide. And this oligosaccharide is responsible for protecting the bacteria from an osmotic shock. So when there is osmotic fluctuation happens in the inside and in response to the outside ionic level, so this is this will help either to protect the bacteria. One example. There is another classic example of another oligosaccharide. That oligosaccharide is made up of about 10 to 14 glucuronic acid. Okay, made up of 10 to 14 glucuronic acid, and these glucuronic acids will join together by alpha 1,4 linkages. So it forms a short chain, and this oligosaccharide is found in plants. What is the function it is doing in plants? So whenever there is a cut occurring in the cell wall of a plant, so there is a possibility that microorganism can get inside. So this oligosaccharide will send a signal. There is a cut has occurred. There is possibility of microorganism entering. So therefore, based on this signal, plants will initiate the defense mechanism to prevent the microorganism from entering. You see the varieties of function that is happening in the oligosaccharides. And we will see, as we take up oligosaccharides one after the other, we will see the various functions these oligosaccharides have in our body. The first oligosaccharide which we are going to discuss is sucrose. So sucrose is the most abundant oligosaccharide in nature. You know that sucrose, we regularly use sucrose in our coffee, in our tea, we add sucrose. It is nothing but we commonly call it the table sugar. It is obtained from sugar cane, sugar beets, pineapple, fruit juice. All these contains sucrose, and it is very sweet. Sucrose is very sweet. So now this sucrose, let us look at the structure of sucrose first of all. So sucrose, as we said, it is a disaccharide. It is made of glucose and fructose. Okay, so these are the two monomeric units of sucrose. Glucose will exist in the D conformation, and it is alpha D glucose, whereas fructose is beta D fructose. Okay, so this is a conformation, and this will exist in alpha 1,2 linkage. So you understand the structure. Let us learn to draw the structure of this particular disaccharide. So what we are going to do is very simple, and this is how we should proceed for all sugars. First, let us draw the structure of D-glucose. So the structure of D-glucose is a 6-carbon sugar, and it is in the alpha conformation. Okay, this is the alpha D-glucose, short form we write. Then we have beta D fructose, and that is in furanose form. Now, normally we give this as the number 1. Now what is going to happen in this case? Remember, if assume this is fructose and this is the number 1. So to draw a bond between here, now this number 1 and this number 1, 2. This is number 2. Okay, number 2. Now to draw a bond between this carbon number 1 to carbon number 2 will become slightly messy. So what are we going to do? If this is what we consider number 2, we will put it close to the number 1. So what we are going to do is, this is the side facing the viewer. We will put this molecule in this order. You see, the entire thing has become upside down. So therefore, if this is going to be carbon number 2, so in the case of fructose, if this is carbon number 2, so therefore, this will be CH2OH, this will be OH, okay, and this will be OH, okay, and this is OH, this is CH2OH, and this is N. This is going to be the structure of beta D-fructose. Now, if the linkage is between carbon number 1 and carbon number 2, a molecule of water is removed, so therefore this is the molecule of water which is removed. By removing a molecule of water, it becomes a glycosidic bond is formed. Okay, a glycosidic bond. This is a glycosidic bond. So a glycosidic bond is formed between either glucose and fructose. What is the nature of this glycosidic bond? It is 1 and 2. There were many experiments carried out in order to prove whether where exactly the bonding is formed. Let us look at some of these interesting experiments. So the first experiment they did was that is in 1950s. The first experiment was they treated this with alpha glucosidase enzyme. So you know that glucosidase will be cleaving glycosidic linkage. What happens is with alpha glucosidase is able to cleave this. Okay, but they experimented also with beta glucosidase. But beta glucosidase was not able to cleave this one. So therefore, it was one indication that it is the alpha conformation of sugar which exists. That was one of the indication. In the second experiment which they did was, they tried to hydrolyze this with sucrase enzyme. Hydrolyze with the sucrase enzyme. Sucrase enzyme again tries to cleave this. Now sucrase enzyme can act only if the fructose is in beta conformation. Okay? Only if it is in beta conformation, sucrase will be able to act. So that was to prove that the fructose is existing in the beta conformation. And the third experiment which was carried out in 1954, then the third experiment is they tried to treat it with an enzyme known as invertase. So invertase again tried to cut this one, and they also added water molecule, but that is heavy isotope oxygen. This is O18. Heavy isotope of oxygen is added, and they wanted to know where it gets incorporated. So therefore, they proved that the 2 units of sucrose is nothing but glucose and fructose, and it exists in the beta conformation, and this is the structure. So therefore, let us name this compound. So we are going to name this compound So this compound, therefore, it is in glucose. This is alpha D glucose, and glucose is existing in the pyranose form, glucopyranosal. There is a linkage, that is 1 to 2 linkage. Okay, alpha D glucopyranosal 1, 2, and the next we have is the beta fructofuranoside. You see how we name a disaccharide. Alpha D glucopyranosal 1, 2 beta fructofuranoside. This is how we name disaccharides. Okay, remember this. Now look at glucose for sucrose for one another important aspect. So let us number the carbon atom. So therefore, this is carbon number 1, this is 2, this is 3, 4, 5 and 6. In the case of glucose, number 1, this is 2, this is 3, 4, 5 and 6. What is the position of the anomeric carbon atom? In the anomeric carbon atom, one was here, second here, they are entering into a glycosidic bond. So the anomeric carbon atoms are not free. Now remember, if the anomeric carbon atom is not free, or it cannot be made potentially free, it cannot undergo or it cannot participate in oxidation reduction reactions. So those sugars which undergoes oxidation or reduction or can participate in such reactions is what we call reducing sugar. So what about sucrose? Though these anomeric carbon atoms are not free, therefore sucrose is a non-reducing sugar, which also means that sucrose in presence of alkali has no effect on sucrose, whereas acid will have an effect on sucrose. So it can be very easily digested, it can be very easily split. This bond can be split in acidic, mild acidic condition. So you know that vinegar is used for degrading this, especially in the confectionery industry. So this is something about sucrose. Let us look at the optical activity of sucrose also. Sucrose is optically active. So therefore, if the optical activity of sucrose is the specific rotation of sucrose is 66.5 plus. This is a specific rotation of sucrose. Okay. Now suppose we hydrolyze sucrose, and this hydrolyzed product is used in the polarimeter. Then you know what happens? Then you have two products. If the two products are, you have D-glucose and you will also have D-fructose. What is the specific rotation of glucose? It is 52.5, that is plus. Specific rotation of glucose is 52.5. Specific rotation of fructose is minus 92 degrees. So which is more powerful? This minus. So therefore, since fructose is more levorotatory, that will overcome the dextrorotatory nature of glucose. So effectively, when it is hydrolyzed, you will see that in the polarimeter, fructose is moving from a plus sign to a minus sign, from dextrorotatory to a levorotatory. Inversion occurs. Inversion. So therefore, sucrose is called invert sugar. So by the activity of invertase enzyme, inversion occurs, so it is known as invertase. Honey contains a lot of invert sugar. That's why it is very, very sweet. So these are the points that you need to remember about sucrose. We have looked at the structure, we have looked at the physical properties, and we have also looked at certain nuances of its chemical reactions, and it's about its abundance, etc. The next disaccharide we are going to deal with is lactose. So what is the first thought that comes to your mind when you hear lactose? It's milk. Okay, milk contains about 5% of milk is nothing but lactose. So it is present in milk. Now, if you look at the structure of lactose, it basically has galactose and glucose. So these are the two monomeric units of lactose. Galactose is a 6-carbon monosaccharide. Glucose is also a 6-carbon monosaccharide. Now what is important for us to know is in which form it will exist. So galactose, let us draw the structure of galactose. So that is how we proceed. Galactose, as you know, is an epimer of glucose. So therefore at the fourth position there is a change, and galactose will exist in the beta conformation. So therefore this is beta D-galactose. Beta D-galactose. Okay, so let us also number this carbon atom. So this is number 1, number 2, number 3, number 4, 5 and 6. Okay, we have drawn galactose. Now let us also draw the structure of glucose. CH2OH. Glucose will exist in the beta conformation. So therefore this is beta D-glucose. Now glycosidic bond is formed between, and this is carbon number 1, 2, 3, 4, 5 and 6. Glycosidic bond is formed between 1 and 4 by removal of a molecule of water a glycosidic bond is formed, we have galactose. So what will be the name of this molecule? The name will be beta D galactopyranosyl. Now beta 1,4 beta D glucopyranoside. So beta D galactopyranosyl, beta 1,4 beta D glucopyranoside is the name of galactose. Now remember, we consume a lot of milk. When we consume this milk, lactose will be there in our intestine. Now lactose cannot be directly absorbed by our system. So this lactose has to be cleaved into its monomeric units. So which are the monomeric units? D-glucose and D-galactose. Now there is an enzyme present, and that enzyme is in our intestine. Enzyme is known as lactase enzyme, lactase. So lactase will cleave these two, glucose and galactose, and it will be absorbed as glucose and galactose, adding to the energy system of our body. So this is the basic understanding about the lactose molecule.